In order for translation to start, we need a few key ingredients first. A ribosome, composed of a large subunit and a small subunit. Notice in the large subunit of the ribosome, the three sites, the E site, the P site, and the A site, where transfer RNA comes to attach to mRNA. The other two ingredients that you need for translation are, as mentioned before, mRNA and tRNA. So, translation first starts with mRNA entering the ribosome, then a tRNA comes to the P site, skipping the A site, and binds to a start codon of the mRNA.
And after the initiation phase, the tRNA enters the A site of the ribosome, and the P site, the TNR in the P site hands over the amino acid chain to the tRNA in the A site. As soon as the tRNA in the P site exchanges the amino acid, it then goes to the E site and exits the ribosome.
The end of translation is the formation of a new polypeptide chain. How this happens is one of the three stop codons signals for termination. In doing this, the synthesis of the polypeptide chain is terminated by a protein called a release factor. And this recognizes the termination codon in an mRNA sequence. Within this process, the release factor bonds to the A site, here, and the bond between the tRNA and the P site, and the final amino acid of the polypeptide chain breaks this translation, separates it to individual parts.
RNA is read by the mRNA, which starts at the promoter, and then it goes along the strand of RNA, and it passes the binding site and creates protein. Now, when an inhibitor is introduced, it goes to the binding site and prohibits the mRNA from reading the whole strand, which results in no proteins being made.
So the PowerPoint says important for cell response to stressors and for growth and differentiation. So for the differentiation, once the protein has been read, it is dependent upon the RNA sequence. So for every RNA sequence, the protein could be different. So that can control how it's read in the codon chart.
So in translational regulation, the regulation of proteins are more sufficient than transcriptional regulation. In translational regulation, the proteins are being made throughout the sequence. However, during the sequence, bad proteins are being thrown out. In transcriptional regulation, the proteins are being made through the sequence. However, at the end of the sequence, the regulation of the bad proteins are being thrown out at the end.
The PowerPoint says that translational regulation controls the levels and amounts of protein synthesized from its mRNA. So basically, the mRNA reads the RNA and is the controlled element of how many proteins or what proteins are being produced.